Hello and welcome to Nikolai's genetics lessons, and in this video we are going to talk about impossible blood group inheritance, and specifically we are going to talk about Cis AB allele. So first of all, I want you to take a look at this pedigree, and specifically I want you try to understand how it is possible for these parents who belong to the blood group A and blood group AB to have child who belong to the blood group O. So from your textbook, you know that we are all belong to the four blood groups: blood group A, AB, B, and blood group O. So those people who belong to the blood group A can belong to two genotypes, whether it can be A or A, and those who belong to the blood group AB would have only one possible genotype, AB. So that means that this person, this male, can be of the genotype whether AA or AO. I mean this person. And of course, if we assume that this person of the genotype AO, that would give us more variants in the progeny. So let's assume that this person AO. More than that, if you take a look at the daughter of this couple, this O phenotype means that their daughter have genotype which is O, because blood group O is made by only one genotype which is O. So again, this is blood group A, blood group AB, and blood group O. And this is genotypes that makes these three blood groups. So now let's assume that this person belongs to the blood group O and has genotype O. That means that this female just have to get one O allele from the father side and one O allele from the mother side. So we know that genotype of the father is AO. But how come that this female got another O allele from her mother? And this is what I'm going to explain you. So according to your textbook, for example, if we assume that mother is AB genotype, and this is what we can see, and so this is mother side and father side. We know that father is genotype AO. And if we build simple Punnett square, what progeny we can expect? And we can expect that this couple may have a progeny that would have genotype AA, AB, AO, and BO. So this couple may have children of the following blood types: blood group A, blood group A, blood group AB, and blood group B. But how come that they have a child who belongs to the blood group O? And this is what I'm going to explain in the next slide. So here you see ABO gene. This gene we can find on the chromosome number 9. And as you see, all the chromosomes in this karyotype arranged according to their sizes, except of course sex chromosomes X and Y. And it also seems like chromosome 21 is smaller than chromosome 22, and this is true. But scientists, before they have been able to sequence all the chromosomes, were thinking that chromosome number 22 is smaller than 21. So this is just interesting fact, but has nothing to do with ABO gene. And of course, as you see, every chromosome here is present in one copy, but actually we have two copies of each chromosome. So, for example, chromosome number 9 would be also present in two copies. Now I want you to pay attention to this gene. So what this gene makes? First of all, this gene consists of 7 exons, which separated by introns, of course. And cDNA, so C stands for the copy DNA, is just all exons connected, so no introns here. And what is the product of this ABO gene? Some people thinking that this is 3 types of the proteins that we call antigens that we can find on the surface of red blood cells, but this is actually not so. As you see, sugars specify actually type of the blood, which we call blood type O, A, B, or AB. And actually this substrate here is naturally found on the surface of the red blood cells. And protein that produced by ABO gene actually is just enzyme. And here is how this enzyme looks like and what these enzymes do. This enzyme just use the substrate, which is polysaccharide, and adds one more sugar. It can add, for example, an acetyl galactosamine, then this would be blood group A. If we have a different allele which would add galactose, then this is going to be to this substrate, then it's going to be blood group B. So this is just enzyme that modifies this chain of sugars. And if this enzyme would be inactive, and this is allele O, as you see, do you see stop codon here? So allele O is just basically inactive enzyme. Enzyme that, as you see, would be very short, and most of wouldn't be even made because due to mutation, it has stop codon very early in its synthesis. So that means that this enzyme wouldn't be produced and the substrate would be unchanged. So we call this O allele. But again, it doesn't mean that these 3 alleles would be proteins which we can find on the surface of the red blood cells. This is just enzyme. So I think this is clear, and again, I want you to pay attention to this picture again, how allele A and B are different. So first of all, this enzyme is coded by 1065 nucleotides. You know that each codon, which consists of 3 nucleotides, would code for 1 amino acid. So 355 amino acids make allele A and B. And what's the difference between them? Just few point mutations. For example, nucleotide 261 in allele A is guanine, in allele B also guanine. And for example, 297 here is at this position nucleotide would be adenine, and here would be guanine. And again, at position 526, A allele would have cytosine, and B allele would have guanine. And this time, if in the first case this change of the nucleotide didn't change amino acid sequence, but this time this change in this in this nucleotide change amino acid. So here we see in the A allele arginine, and here we see in allele B glycine. And we also have another point mutation here. Again, it doesn't change amino acid sequence, but this change does change amino acid sequence. But again, compare allele A and B, actually just has, let's count how many different amino acids. For example, we have one change of the amino acid, second, third, and fourth. So just 4 amino acids out of 355 would be different between allele A and allele B. One more time, it is very important to understand that basically we have one enzyme which, if it is A allele, would add an acetyl galactosamine to this sugar chain, and if it is B allele, would add galactose, as you see. And those people who belong to the blood group AB, they have basically just 2 different alleles. One inherited from the, say, mother side, allele A, and another from the father side, allele B. So on each red blood cell, they would have 2 variants of these sugar chains. One would end with an acetyl galactosamine, another would end with galactose. And this is going to be blood group AB. Now we can return to our original slide. So what Cis AB means? In chemistry, for example, if we take some molecule, cis means that we have on the same side 2 R groups. And trans means that we have R groups on the different sides. For example, here and here. But we cannot apply the same rule to genetics. We have double-stranded DNA. And what do we mean by Cis AB? And let me first show you something. So this is chromosome number 9. And we have 2 such chromosomes, number 9. As you remember, one we inherit from mother side, another from the father side. And ABO gene would be somewhere close to the tip. And now let me take this gene and increase the size. So what happens here to this gene? So let's now say this is gene. And you know that many chromosomes would during meiosis cross over, and crossing over may happen in many places. Usually it takes a place outside the gene. Why in most of the cases? Because genes only 2% of our genome, so 2% of the chromosome would be sequence that code for certain proteins, and the rest 98% would be non-coding sequence. So when we have crossing over, usually it happens outside the gene, outside of the exons. But still, sometimes what might happen, we may get a crossing over that happens in the middle of the exon. So let's say it happened here, and what we can get? We can get recombined exon and recombined gene and new allele. As you remember, allele A and B, and let's say this is allele A from the mother side, and this is homologous but not exact copy of the same chromosome, and it has B allele here. But this crossing over would give us a new variant of this allele. So let me use different color. For example, this arm now would be here, and this arm would come together. And as you remember, allele B and A, difference between them is just 4 amino acids. And let's assume that result of such a crossing over would be new allele that is going to have only, instead of 4 differences between them, just 2 differences. And what's going to happen? Such enzyme, and this is enzyme, would sometimes convert substrate into one type of polysaccharide, or add an acetylgalactosamine, and this is going to be A blood group, and sometimes would add another sugar, which is galactose, and this is going to be B allele. So now this enzyme, or this gene that code for enzyme, would use a polysaccharide that is attached to the surface of the red blood cell, and sometimes would add an acetyl galactosamine, and sometimes would add galactose. So this means that this person would has both alleles A, or antigens A, and antigen B. And we can say that genotype of this person would be AB. But this would be specified by only one chromosome. Now let's imagine that we have some person who has such chromosome with such allele, which we call Cis AB. So let's say this person inherited from one of the parent such Cis AB allele, and another homologous chromosome inherited from the other parent in the same locus has O allele. So what does it mean? That means that enzyme wouldn't be produced at all. Now let's return to our pedigree here, and let's take a look at what's happening here in this new light of our knowledge. So we know for sure that this male is blood type A O, because the daughter have to get one O allele from one parent and another O allele from the other parent. So take a look what actually happens here. So male parent is genotype A O, female parent is genotype A B O. Again, it doesn't mean that this parent, female parent, has 3 alleles. This allele, which is Cis AB, can produce enzyme that sometimes would make add one type of sugar, sometimes add another type of sugar to this polysaccharide. And still this person has just 2 alleles. One is just Cis AB. And now let's build a Punnett square, and let's take a look what's going to happen in their progeny. So one parent can give A allele, another AB. So what is going to be genotype and phenotype of this person? It's going to be A, B, A. But basically that means AB. Let's take a look what we are going to get in this cell. A allele progeny can get from male parent, and O allele can get from female parent. So this is going to be blood group A, and AB from the mother side and O allele from the father side. So what would be phenotype? Phenotype would be AB. So this is going to be blood group AB. And again, we also can see in the progeny that one parent can give allele O, and another parent also can give allele O. So in the progeny we can see impossible phenotype, or again, according to this pedigree, when one parent A, another AB, we do not expect that progeny would belong to the blood group O, because only one parent, as you see, can give allele O, but we do not expect that this parent can give allele O. We expect that this is going to be whether allele A or allele B. So we can expect that progeny only can be whether blood group A, AB, or blood group B. But again, we do not expect that progeny would be blood group O. But in the light of this new knowledge, now you know that sometimes we may have such alleles that actually produce such enzyme that can actually work like both enzyme A and B, and add both types of sugars. And again, this explains what we see in this pedigree. Again, cis here stand for the not two alleles on one chromosome, but that one allele has dual function. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.